So that's why we brought these two people requested, and thank you, Dr. Abhay, Dr. Professor Bharatan, for being with us to spend some time and induct the interdisciplinary science and their built-in facilities into the young brains and wonderful brains of the NIPER library. And science in the sense, science is there if you observe with the sense. The moment the child born, whether it is a boy or girl, there is a sound, some sound will come now. If you record that sound and then translate it to the oscilloscope, you will get that oscillograms. Now everything is automated. The sound, if you touch it, put it to the computer, you will get the peaks. That means there is a what is that wavelength is in between the two peaks. There is a distance actually, the megahertz that can be calculated and all. So that means that sound is a kind of a species specific, gender specific. That means science started with your born itself. Okay, then when you are crawling, you will be feeling happy. Oh, my son is crawling. After that, he is walking and all. That means kinetic energy is there in that. That means boy is spending some energy to make a move from A place to B place to cover certain distance. That means right from the born of the child almost till last breath, science is a part of our life. It is up to we people how to exploit the science. If anyone want to talk anything about the, I already told earlier also about any kind of a possible solution for the available diseases, the existing diseases, that is only from India only. Because phytopharmaceuticals and biopharmaceuticals, we are talking the many, but we are only supposed to talk. It's not the what do you call ego or pride. Why I am telling, I have a lot of material here in the country. Huge biodiversity is there. Then excellent chemists are there. Exploit the chemicals. It may not be overnight story, may not be tomorrow, may not be after decade. But once you get a molecule, it will cover the backlog of 30 years and then see the another 100 years of the future to counter the diseases like we never had orphan disease, we never had a rare disease, we never had a re-emerging disease, we never had the emerging disease. To do all those things, we need more chemists, we need more biologists to understand the disease biology. Please understand this. When you are talking about even your molecule targeting the cancer, first understand the disease biology. Then only you go to the your either chemistry side or biology side. With these few words once again, welcome all of you, and let me congratulate once again and my wishes to all of you on this wonderful occasion. You know, Sir C.V. Raman, whoever goes to Bangalore, you should spend some time to see the C.V. Raman building. In that, with his handwritten, he has written some quotations. You should read that, you will be excited and you will be emotional. So, one should, I told you the word of ego just now. Somebody asked Sir C.V. Raman, sir, you got the Nobel Prize in Bharat Ratna. How you are sustaining this ego? He told very lighter way. No, that is why ego should not touch my head. That's why my head is tightly tied with the turban. No ego will come and touch it. These are all the simple things. That means how one should avoid the ego also. He has nicely narrated and documented with his own handwriting. So smallest things are there. It matters a lot. Yeah, after all, 1 2 publications, if you publish, you will raise the collar. There are so many people. Above you see them and learn from them, and be like seriously, be like isomers of at least C.V. Raman. At least I can see 0.1% of this population look for the science. That itself is a great contribution to the great country and a great nation. Thank you very much. Jai Hind.
Thank you, sir, for your kind words, and definitely these words will motivate our students as well as their inclinations and interests. It will be raised up in their science. And now we are grateful that we have 2 speakers between us, Professor P.V. Bharatam and Professor Abhay Pandey. So first of all, our first speaker, Professor Abhay Pandey, I would like to just little bit introductions among you people, because I think most of us, we are aware about his contribution in science, especially in protein engineering. So Professor Abhay Pandey sir ji is senior faculty in the Department of Biotechnology at National Institute of Pharmaceutical Education and Research, NIPER, SAS Nagar, Punjab. Professor Pandey has more than 26 years of experience in the field of protein research, and his primary research interest is protein engineering and development of protein therapeutics. He has published more than 43 papers in the national and international peer-reviewed journals and has filed 7 patents. He was a part of team receiving prestigious CSIR Technology Award for Life Sciences in 2018. Currently, the lab is engaged in the development of novel proteins therapeutics for clinical use. In this presentation, Professor Pandey is going to talk on various aspects of biopharmaceuticals. He will also share his research on the development of human arginase for clinical use. So now I request Professor Abhay sir, please deliver his talk among our students.
Thank you very much, Dr. Niti. I would like first of all to extend my warm wishes on this very auspicious day, Science Day, National Science Day. We are all scientists or science students. So this is a big day for us. And when Professor Murthy asked me to give a talk, I readily agreed, and of course Professor Bhartam also. And this also gave us an opportunity to interact with all of you. Because of past 2 years COVID pandemic, life was little bit restricted for all of us. And now, since we are getting an opportunity or time, so we took that opportunity to come out and to interact with our family members. As Professor Murthy said, we are the family member. It doesn't necessarily means NIPER R, NIPER M, NIPER A. At the end of the day, we all are on the same board. So with that word, I thank you all for giving me this opportunity. And today I will talk on 2 aspects. Okay. I will spend some time on biopharmaceuticals, a very emerging discipline which our nation is targeting. And one of the segment in that is a protein, of course. And in the later part, I will share with you some of the data of one of our program in which we are building one enzyme, human enzyme. For so, India is the pharmacy of world. We should not forget. Whenever there is a crisis, world look at India. Moving Indian pharmaceutical industry is a leading source of path-breaking innovation. So our pharma sector is very powerful and is a leading source of path-breaking innovation. Currently available major segment of our pharmaceutical industries, all of us are aware: generic, API, intermediate, drug formulation, vaccines, biologicals, Ayush herbal, surgical and medical devices. So this is the right time to ask a question: can we put protein biopharmaceutical also as another major segment in the arsenal of our pharmaceutical industry? It is pertinent to remember our pharma vision of our government of India 2020 is to make India a major hub for end-to-end drug discovery as well as global leadership. Unless until we realize our job duties, that is very important for us. We will not move in that direction. Protein biopharmaceutical, what are they? Are mostly recombinant proteins. They offer many advantages over chemical synthesized small molecule drug. So we biology want something to say about chemical drugs. So we say that okay, ours is better. But in reality, all drugs are drugs. But yes, protein biopharmaceuticals are gaining advantage over chemical drug because of many things. I will not go into detail of that. Just one survey I will talk about 2018. This survey is done every 4 years. As per this survey, 207 products was approved by US FDA, one of the authority in the drug approval, which contain chemical or biological entities, and that was for 4.5 years between this period. So I'm waiting for the next survey, which is supposed to come in 2022. We will see what is the status there. When I analyzed that survey, I found 97 products, that is 40% of this, were biopharmaceutical. And this increase in the percentage, 47%, was substantial from the previous survey, which was 26% and 20% in the previous 4-year surveys. So this is an indication that biopharmaceuticals are gearing up, and there is no denial of it. I think we all accept that. When you look into the product, major segment is still a monoclonal antibody, followed by hormones and other proteins-based drugs. So when you calculate, it comes out to be 94% of the protein of the product biopharmaceutical are protein biopharmaceutical. So when we say biopharmaceutical, as per this data, majority of 90% of it is a protein. Okay, so protein biopharmaceutical, what are they? They are recombinant protein for therapeutic use. There are 3 categories. Biosimilar, a word which has come to us many times in many different forms and shapes. Biosimilars are nothing but similar, which are not identical to the innovator molecule. If somebody has made one discovered a new protein for therapeutic use, and the other can make it with certain rules, then those molecules are called as biosimilar. They are also called by various names: follow-on biologics, biogenerics, similar biologics, and so on and so forth. For many of us, these terminologies are very confusing. They are only biosimilars. They are still in demand. Next category, which people have moved after biosimilar, is called as biobetter. They are better version of the biosimilar. For example, if you made a drug and which is not able to cross a blood-brain barrier, it is still an effective drug, but it is not crossing blood-brain barrier. And if you improve by protein engineering and allow it to go into the brain where it is needed, then those molecules are called as biobetter. It will still do the same job, but it has got the added property. So biobetters are those one, improved or upgraded version of innovator molecule. They are also called by a variety of names. And this is actually currently in one of the fastest growing segment in biopharmaceutical market. There are many molecules which are available which need upgradation. So when Windows came, very oldest version, they had some feature. Every 2-3 years, the version is upgraded with additional property, doing the same thing. It is not doing something different, but with an additional property. And every year we have to update our those kind of operating systems. Similarly, for protein, if you upgrade it, the market is open. And novel proteins are, of course, they are always there. They are new drug molecule, and we all know they take a lot of time and money to develop. So biosimilars nowadays, except for monoclonal antibodies, have become a low-hanging fruit. They have become a low-hanging fruit. You will spend same energy, but the price or market value is not that much. If you invest a little bit more, if you increase any of the property, then you are in the game. So I concentrated here addressing this issue. Many approved and developing proteins therapeutics, they need improvement in their variety of properties. These are the 4 major ones which I understood. Suboptimal pharmacokinetics. So we have a very good drugs, but their half-life is less than 2 hours or 1 hour. That means we have to continuously infuse it at the cost of it. If you increase the half-life, you are in game. And that is the biobetter, and the market is open. Similarly, suboptimal pharmacodynamics, poor blood-brain barrier properties, and poor target specificity. So these are the sections available to target to make a biobetter from the existing biosimilar. They will still do the same function, but with the improved properties, they will be much more clinically used. Many drugs, most of the approved and developing drugs, they have a half-life of less than 1 day. Many in the range of minute to few hours. We have a very beautiful, very good efficacious protein molecule, but their half-life is less than few hours. So they need an upliftment or upgradation, and if you do that, you are in business. Of course, all 7 NIPERs are engaged in high-end biological research. I understand and acknowledge that, and thank you very much for NIPER also for their contribution in the NIPER research in different disciplines. And these disciplines or their expertise complement in the development of biopharmaceuticals, or can be made complementary in the development of biopharmaceuticals. So that was in brief about biopharmaceutical. Now I will talk about research programs in my laboratory, which is going on for 15 years. I have to summarize, 4 research programs are going on. Anti-cancer agent development, where we are developing a human enzyme, arginase enzyme, for clinical use. In another program, we are developing a prophylactic enzyme as a nerve agent, prophylactic against nerve agent poisoning. In another program, we are developing a small biologics for inflammation, and bispecific nano antibody is the last program which we have conceived for past 3-4 years. So bispecific means this antibody can bind to 2 antigen. This can quench 2 inflammatory markers. TNF-alpha and IL-23 I am targeting. As we all know, COVID, the second manifestation of COVID is a severe inflammation, uncontrolled inflammation throughout the body, and that has caused much damage to the infected person. And in that, TNF-alpha and IL-23 both axes were heavily inflamed. So if we can give this protein which can quench both, it can be a very powerful anti-inflammatory. But for this talk, I will only talk about arginase journey. So development of human arginase, albuarginase as a broad spectrum anti-cancer agent. Well, what is arginine? Important amino acid, all of my dear students, they know, play vital role in physiology and pathology. However, its metabolism is altered in certain diseases. In such cases, if you administer arginine hydrolyzing enzyme, that provides a beneficial effect in these conditions. So if you give an injection of arginine hydrolyzing enzyme, which is an arginase also, you can address variety of diseases like arginase deficiency, oxotropic cancer, immune system, and other things as well. I am concentrating on this only. So arginine hydrolyzing enzyme, they have emerged as a strong candidate for treatment of variety of disease and conditions. So what is arginine role in cancer? Arginine oxotropic cancer. Certain cancer cannot synthesize arginine and depend on the body pool for their growth and maintenance. So when a normal cell becomes cancerous cell, it adopts some good property and loses some good property. It becomes uncontrolled, dysregulated with respect to its multiplication, but it also becomes voracious. It needs a lot of ingredient to multiply quickly. In that, arginine is also one of the amino acid it needs in large amount. Ironically, that amino acid capacity, synthesizing capacity is also lost in that. So it depends on body pool of amino acid. And if you cut this arginine, if you don't give food to the cancer, cancer will be under stress. So normal cell can synthesize amino acid, arginine, while cancerous cell, they cannot synthesize. On top of that, they need lot. So if you neutralize or if you degrade or if you lower the concentration of arginine by giving enzyme arginase, the cancer cell will be stressed. Then that itself can cause growth of cancer to halt. If you add chemo, again the second weapon, that will be more lethal. Or if you here target by autophagy inhibitor or by checkpoint inhibitor or by apoptosis inducer, which can initiate or amplify the effect of this arginine inhibition in the cancer. So point here is different companies they are targeting. Now we know arginase is important for cancer, and there are very many different cancers. Some companies developing a new combination with chemo. To protect the IP, some companies developing with the autophagy inhibitor against particular cancer. Against another set of cancer, they are developing in combination with checkpoint inhibitor. So it has opened a whole new spectrum, and whoever will get a result will come into market. For these 2 enzymes are established, arginine deaminase, it is from bacteria, and of course human arginase 1, they are undergoing clinical trial. So I will just quickly go through the players which are already present in market.
arginine deaminase. It also hydrolyzes arginine. It is a bacterial enzyme. Polaris is developing this enzyme as a pegylated version. I hope all of you know pegylated version. Pegylation of protein impart additional properties, resistance to proteolysis, and so many things. So this pegylated version is in market by Polaris. Interestingly, it is being developed for a variety of cancer. So this is a broad spectrum anti-cancer agent. It is not for one cancer. For example, you have a monoclonal antibody that will only target pancreatic cancer. So this is not a case. So if you develop a molecule, then you will have a whole field open. This molecule received orphan drug status for liver cancer and invasive malignant melanoma. However, this enzyme suffers from limitation. This enzyme converts arginine into ammonia. So patients have hyperammonia syndrome. So ammonia toxicity is being reported. Also, the enzyme is microbial in nature. We generally prefer human enzyme. If it is there, we want as human as possible. So microbial enzymes are easily being substituted if you have a human counterpart. So it is highly immunogenic. Adverse drug reactions are very common. Also, PEG toxicity is an issue. So that comes human arginase is an alternative one. Fine. This enzyme hydrolyzes arginine into urea, so no ammonia toxicity. Injection of this enzyme inhibit growth and progression of many cancer. So that is a very good property we want. However, the enzyme has low circulatory half-life, 4 hours. This is the challenge I took when I started this program. I got the molecule, okay, enzyme is working. We produce the molecule, but its half-life is less. Now came my part. As a protein engineer, we entered into that. Before that, let me give you a picture of the market. Realization of this, 3 pegylated versions are being developed, 2 by Chinese company. So these are pegylated versions. And one is by US company. So they are in market. The race has begun to who will come to first, and all phase 3 studies are going on. This takes time. The development obviously takes time. But if you have molecule in pipeline, that gives you hope that it will eventually come to some stage. So these 3 molecules are pegylated version of arginase, and they are under development. As I said, this enzyme drug is being studied with the other molecular targeting or cytotoxic agent also. So variety of chemos are being tried in combination. Different companies are trying different combination for different set of cancer. Autophagy inhibitor and monoclonal antibody as well as other drugs as well being tried in combination. So this is the biggest area for IP generation, and companies are battling very hardly to find and establish their niche. This enzyme shows a very good anti-cancer effect in variety of preclinical models, preclinical studies in different cancer, as well as in clinical studies. So we can see a whole spectrum of cancer can be addressed by just one molecule. If you have one molecule, then the market is open. So this is a broad spectrum anti-cancer agent. This molecule will be different than the monoclonal antibody, which is a specific cancer. If you have one, you will take one and that will be effective against that particular cancer and not the other. But this molecule can address this issue. However, since we are developing the non-pegylated, we have to find some lacunae in pegylated. Of course, they are there, we know that. But as a separate rival, we have to throw some dirt on that. So, PEG toxicity is recognized as an issue, we are saying, and it is known. Nowadays, people are moving away from pegylation. Because a lot of PEG accumulate in the body. PEG is non-biodegradable. And in the organs, it accumulates and causes all kind of problems. So there is a dire need to develop a safer arginase for addressing this circulatory half-life. Using protein engineering approach, we tried to address this. Also, a recent analysis by me, I went into the clinical trials and found that companies are moving away from the pegylated one. Pegylated one is given in the green, and these are the engineered or fusion protein one are given in the blue. So the percentage of pegylated protein being approved or in the clinical trials is much less than or substantially less than the fusion counterpart. So in the international scenario, this is where we can place our technology. We have 4 players ahead of us at different phases, but they are all pegylated version. And we thought if we develop a non-pegylated arginase having a desirable property, then it impart following advantages to its rival, which is there in the clinical development. Okay, so what is fusion protein? It is used to increase the circulatory half-life of drug. Pharmaceutical companies are using this approach to develop biobetter. More than 13 fusion proteins are already approved by US FDA. Fusion proteins are better than pegylated proteins. So it is known. What happens when you give the protein? There are multiple mechanisms by which it clears from the body. One of the major mechanisms is a renal filtration. So glomerular filtration, if protein size is less than 60 kD, it will filter through like a seal. If you increase the size of protein, it will not filter in first round, it will have one more round in the circulation, and that way it increases the half-life. So fusion asparaginase, a golden anti-cancer drug, which is working for 50 years. This is a cousin of arginase, is being used for more than 40-50 years. And fusion ADI are also being developed. This is the basic construction of fusion protein, our drug molecule, and a component, second component, to increase its half-life or targeting cancer or blood-brain barrier permeability or whatever you want. And this need to be fused in a unison with a, I prefer to be done by a random, by a peptide linkers rather than the chemical joint. So these are the 3 components: drug, linker, and helper molecule. Okay. These are the stages we started with. In the stage 1, we designed screening and identification of lead variant. Since we are designing the protein de novo, we do not know. We are creating an artificial protein. We are creating a chimera, attaching fragment of this with fragment of that. Whether it will express, whether it will be active, whether it will be stable, we do not know. So that screening was done and followed by other steps. So the panel of molecules are designed with a drug, linker, variety of linkers are being used, and the other partner. And out of those, 13 variants were cloned. That was enough for me. Based on my wisdom, I cannot do 70 molecules, generating 70 protein, cloning, expressing, purifying. It's a humongous task. So we narrowed it down based on our wisdom, whatever our knowledge tells us. 13 molecules we cloned, expressed, purified, 4 lead variants which were coming in conclusive way in our lab. We purified and compared their activity, and we got the 2 variants which are consistently coming in our hand and are showing a desirable enzymatic activity. They are trade name albuarginase and transarginase. So that was our first step. We got the lead molecule. And the second step was to develop a process for its production. Obviously, once you have small amount of anything, the second step will be how can you develop a process so that you can create a large amount. We use industrial viable approach, that is using Pichia pastoris. Pichia is a yeast expression system which is used for producing many drugs, recombinant protein, which are available on our shelf. We have to move it from E. coli to Pichia because this is more industrial relevant. Because we have to sell it to industry, industry will grow, so we have to produce in their skeleton only. We developed this process, and a novel lab scale process is being developed, and we got a good clones, and we are improving, keep on improving the process for producing high yield of protein. Then we did the biological characterization of one of the lead, albuarginase. This was done with Professor K.B. Tipu, Department of Pharmacology and Toxicology, another expert in the field. Half-life determination in rats, PK studies of this molecule suggest that it has many-fold greater than unmodified arginase. So this is substantially first step. We got a good result that earlier unmodified arginase is a 4-hour, and as per our calculation, we are getting substantially higher. I will not disclose the amount. I have to file a patent, so that is why the specific data are not. We believe that this is a renal filtration because we have engineered a new molecule is doing this way. or there is a FcRn mediated recycling may be responsible for that. So this we will prove eventually once we will come to the next stage of the program. Anti-cancer activity against the cell line. These are representative. We know it is going to act, but just to make sure whether it is acting or not, we have checked it against representative cell lines, and we got that it is showing the activity which is consistent with the values reported in the heart for the coagulated one. As a representative anti-cancer activity in vivo, we use the xenograph model of SCC, and we found that albuarginase in combination with the 5-fluorouracil, another standard one. Actually, we haven't decided finally, unfortunately, which combination we want. I myself is confused. There are so many options available. Whether I should go for pancreatic cancer, whether I should go for lung cancer. Because once I am committed, I will be committed for 3-4 years. Which combination shall I take? Shall I take cisplatin? Shall I take this? Shall I take that? Or shall I take new one? Those which are having in another lab in India. So all these commitment we are yet to make, and this year hopefully we will settle down for core cancer and we will start our journey in a full aggressive way. It is showing, the chimeric molecule is showing what we wanted. So this is a net result. Till now, 2 lead molecules we got. We got a clone for its production and lab scale process. So if anybody wants it, we can give them. This molecule exhibit desirable activity, and as of it now, the TRL level is 3 to 4. Second molecule we are in process of developing. That's another PhD. She will start working on the second molecule. Potential application we anticipate, yes, very many cancer. As I said, along with chemo, I do not know exactly which is the market requirement, which cancer I have to target, or which combination I have to target, or with any other cytotoxic agent. Okay, so this is how I see my journey ahead. We have covered this part, engineering of variant. Second target will be to produce in the gram quantity. Single digit gram quantity I am targeting this year, either by collaboration or by outsourcing. So once you make any new chemical, you have to produce in large amount so that it can be used for the real multiple studies. That is our target for second this year. Simultaneously, we will decide exactly which disease model, which is a market requirement of India or the nation, and which combination is needed. Once that is done, we will execute. We will see whether it is showing desirable effect or not. Selected data or the setup will be used for preclinical data generation, because that's altogether different mindset and story. Generation of preclinical data, either collaborative or by service provider. Our goal is to target to file IND filing maybe 5 to 7 years. With the gracing of God, if it helps, we are targeting IND filing in 5 to 7 years. Journey of mind. I will be happy if you have any questions.
What are the selectable markers you decided for this process?
The selectable markers come with the standard plasmids. So plasmid have their own gentamicin resistance. These are set up things. All these things are already available commercially. Yes.
This was the de novo synthesized protein synthesized, especially that might be that you have had some changes in that marker also, because that is screening process will be more helpful.
No, screening process we use for production only.
Only production.
Yes, molecule we have designed randomly, not randomly based on our wisdom, whatever has permitted us.
So this is totally not, since this is a new one compound that you have synthesized new de novo markers. Once you are introducing inside that, technically it will act as a foreign molecule, like as a xenos for that specific. So, have there any other type of the response against to this, because definitely this molecule should not properly work, and some other markers they will immediately show their response in a negative way. So, is it the?
We have not looked into it. Our idea here was we have.
How long it can retain into the body of that thing, because once we will introduce it in the particular.
24 hours to 35 hours. 35 hours was the T-half we got. So that was a pharmacokinetic study, circulating output. Once we make a molecule, our target, if I made the tea, my first thing is whether it is okay or not. Then comes other things, whether the chini is there, milk is there, you know. Then comes whether flavor and which cup I have to take or other parameter. So right now I'm that stage. Whether it is active or not, if it is active, is it showing a combination effect with the chemo or other agent? So that was my first task. I cannot address everything at one go. Yes, so we have taken 3 years dedicated. Once we reach here, all the things will be addressed. Once we have a particular cancer or particular scenario which is industrially relevant or the field relevant in our hand, then obviously we will spend 3 years in addressing. It need to be, otherwise we cannot go here.
Yes, if you ever thinking that it might be that it will be in your mind, because before deciding that path, definitely some drawbacks we will have to consider it.
Yes, they are known, but these are too far from me. Right now I am here, so I have not started thinking of those things as of yet.
Sorry.
Yes.
Because as you know, and very with respect to cancer, I can think of one of the components. Did then substrate for this antigen.
And no,
That's a very important role in manufacturing.
Yes.
Or the dead cell. Yes. So when we are decreasing that antigen, actually, how do you think, like, once there is a about how those dead cells get of this compromised production?
Yes, that is a very good question. NO hydrolyzing activity of arginine is being used to suppress some of the extremely hyperimmune systems. So it is being developed for anti-inflammatory also. And when I look, they are saying that it is cutting the arginine and not providing substrates to generate NO. Right. So if less NO is generated, inflammation will subside. So this molecule has a many role, not only in cancer, but in other disciplines, which I am not doing right now. It's too much for me. Even for cancer also, had I have only one cancer, I will be happy I am targeting particular cancer.
Yeah.
But now,
based upon that, I mean, because nitrogen side play a very important role in formation of melanin and melanogens. I think like these are better for this melanocyte or
yeah, skin cancer is also, yes.
melanoma. Especially, I think, with respect to metastasis, it should be better. Then itself metastasis and that time.
Yes. And always remember, normal cell can make a heavy arginine. So its normal function is not compromised. Normal cell can make its arginine. वो happy है अपना बना के is happy. But the cancer cell, they need more and they cannot make it. And if you stop them from eating, then they feel stress. This is called HNS also, and it's established. There is no doubt in that. Now the race is who develop arginine, who develops combination, and who comes to market. So this is one of the journey of one pharmaceutical. You can come to me anytime after this seminar is over also. When you go home or next week also, if you have any question, I am accessible by email also. As a student, I will be happy to answer your queries.
Yes, they may interact separately also. Thank you, sir. Very informative talk, and I hope that those are who are interested to complete their work in protein engineering, they could get a idea and they can interact further with us, because this is the most important part, that is NIPER Mohali. That is, we can say that grandfather, or overall we all belongs to the same family, but like as a grandfather, grandmother of our NIPERs, so we can. So definitely they can access in future also. Thank you so much. Now I would like to. So now I request Dr. Ava Sharma ma'am, please introduce our next speaker, and that is the next part of today's event. Sorry. Thank you, Dr.
Our next speaker is Professor P.V. Bharatram. You all know he's a professor in the NIPER Mohali, in the Department of Medicinal Chemistry in NIPER Mohali. He's a very well-known researcher and teacher. So as we are from NIPER, so many of the students maybe know sir very well. And I think medicinal chemistry students must be knowing this sir. So he has lot of research and teaching experience many years, and his research interests are synthesis of computationally designed molecules, and in the medicinal chemistry, he is working in the area of anti-malaria, anti-diabetic, and quantum medicinal chemistry, pharmacoinformatics, drug metabolism, drug toxicity, drug delivery. He has a recipient of many awards and honors, Fellowship of Indian Academy of Science, Fellowship of Andhra Pradesh Academy of Science, OPPI Scientist Award, Renmexi Research Award, Fellowship of Royal Society of Chemistry, IBM Faculty Award, and many more awards and honors. These are few I have mentioned. So many. And he has published more than 258 research papers in international and national journals, and he has supervised so many students, PhD scholar, and the figure is around 29 completed, and still 7 are they are pursuing their PhD under him. And he has a huge editorial experience in many journals, few I can mention, and these are guest editor for Indian Journal Chemistry section, and for Springer, one of the this journal, and Indian Journal of Chemistry and current computer-aided drug design, and many more is there. He is a member of national international assignments, and for example, Research Council, CSIR, CDRI, and for member CSIR Chemistry, HRTG Committee, PAC, and so many, like, means so many, you can see. You see me. With small words, I would like to invite Professor P.V. Bharatram sir to deliver his talk on this occasion, and the title of talk is Field Thinking in Anti-Malaria Drug Discovery.
Nice to talk to all of you. It is not audible through my card. It's not easy to get such opportunities, especially during this pandemic period. There is no way of traveling and interacting with young people like you. So it is a good opportunity to share some of our experience in medicinal chemistry. Here, my title is somewhat strange, not a regular title. The idea is that we have got to think in 3 dimensions. We always think in 2 dimensions because whenever we studied right from our childhood, our teachers asked us to write on paper, and they teach using blackboard. So it was always 2D. Teacher was teaching on blackboard in 2D diagrams, and all the figures that we make, even in biology, when we make beautiful figures, they are all 2D figures in the end. So we are somehow trapped in the idea of thinking in 2 dimensions, in chemistry as well as in biology. Now the facilities are available. Facilities are available starting in 1993. Molecular graphics topic has grown. Still, many of our books are written in 2D. We don't use the 3D books. This is a pity. I think somebody should write 3D book in drug discovery. Maybe it is my responsibility to do that. I still have to do that. So we have to think in 3D. Drug discovery cannot happen in 2D at all. So this is a very important message. It is not only given by me, it was a recent excellent article published in the Journal of Medicinal Chemistry. The title of the article is "What Makes a Great Medicinal Chemist?" The clue, one of the clues, is that great medicinal chemists all think in 3 dimensions. This is one of the answers. So I am not giving my answer only, I am giving world perspective. The great medicinal chemists all think in 3 dimensions. So I am inviting you to start your journey today. I am going to give example in the form of our anti-malarial drug discovery. Most of you know about malarial drug discovery. Lots of things are happening. I am going to take one example. Before we do any research, what we do is we look at the macromolecule in 3 dimensions. There are facilities available. PyMol software is available. You can download it free of cost. Every laptop can have it, and every pocket top can also have it. Why not? There are facilities available. Why we have to look at it in 3 dimensions. Rotate, zoom, represent in ball and stick model, represent in wireframe model, represent in this kind of cartoon kind of models, and represent in many ways. And we understand each time. That is why we are in research. Okay. After doing that, we can go ahead and carry out new modeling effort that is based on structure-based drug design, or the pharmacophore mapping approach, or the QACR, 2D, 3D, always 3D we do, and molecular dynamics, always dynamical state of the molecules, understand them, and then go to quantum medicinal chemistry. After doing all that, we go to the designing of the molecule. Once we design, we go to organic lab and we synthesize them, and then we take the help of in vitro characterization, and then we go to the next step, that is in vivo, and that's how research goes on. I want to start with a very small molecule. This small molecule is imidomide. It's not a drug, but it is one of the small molecules. This molecule we try to understand. What is there to understand this? Then how this technology will accept application? If there is nothing, there is so much. So much inside the molecule. Every molecule has got an inside story. Try to understand the inside story. Okay, our friends, all of them have got their internal details. They don't tell us. They have their own what happened in B Farm College. So what kind of things they have done, dynamical things. They don't tell all of them, but they are having these internal secrets. Exactly like that, every molecule has got internal details. These internal details are because of their electronic character. Electrons, molecular orbitals, charges, the second order delocalization, primary delocalization, proton affinities. Likewise, there are so many details with reference to each of the molecules. That is what we studied for many years. This is only a small list that is included here. So these are small molecules. Urea, thiourea, we know them for the science. Since the day we learned about chemistry, we are knowing about these molecules. They are small. They have got several secrets. Now, if you know these many secrets, let us extend a little bit. Guanidine, we understood. So can we work in bigonides? Simple question was asked in 2004 by our team, and I asked one of the master student like you, why don't you work on this molecule, metformin? He said, sir, this is a 50-year-old molecule, everybody knows about it. I said, okay, if everybody knows about it, don't work on it. Don't do it, right? Because everything and anything about this molecule probably is already known, so don't work on it. But the student, second year master's student, first month of his research, he told, sir, I want to work on this for 2 days. Okay, I said, okay, tell me, take 15 days time. After 15 days, come back, tell me that I have wasted, sorry, I wasted my time. I will take up another topic. Tell me this answer after 15 days. I am giving you free time. After 15 days, he came to me and told that, sir, I did not waste my time. Master second year student, he was telling something against his own mentor. Okay, telling that I did not waste my time. I want to continue to work on this topic. Then I asked him, why are you telling you did not waste? That this structure is wrong. The structure of metformin is wrong. Then we looked into Journal of Medicinal Chemistry. All papers published last 15 years. This wrong structure was written by everybody. Structure of metformin was wrong, buformin was wrong, chlorhexidine was wrong. All the structures are wrong. That's what he told. Second year master student of NIPER has the guts to tell entire Journal of Medicinal Chemistry 50 years of papers are wrong. That is NIPER capacity. NIPER students, all of you have this much capacity. Do not hesitate. Okay, this is what he told me. And then I went to lab and I saw what is this, why is he telling like this? And then we realized that he is right. But this structure is indeed wrong. What is right? This is how you write it in full. It's a green structure, structure number 1, it is wrong because this is the wrong tautomer. Simple chemistry, every day we learn about chemistry. Tautomerism is known to us. This structure is wrong, this structure is right. What is change? Only 1,3 hydrogen shift. So, but what is its implication? It's a huge implication because the energy difference between these two is of the order of 9.3 kilocalories per mole. 9.3 kilocalories. If you convert into Boltzmann's formula, then that formula is N1 by N2 is equal to delta E to the power of epsilon to the power of delta E by KT. Simple Boltzmann's law. And so use that, it becomes clear that if you have million molecules belonging to this stage, only one molecule will have this structure. This tautomeric state is followed by billion. Thus, this wrong tautomer is followed by only one. So then why do we write the wrong tautomer in all our publications? Let us don't do that. This was the message. Then over and above that, if you write the structure of trigonide in this way, you will have this kind of resonance stability. Extra stability due to resonance, that is a hydrogen bond assisted aromaticity is helping in hopping of the hydrogen on both sides, and thus this molecule has got extra advantage. Okay, so then this is the 3D structure of the right tautomeric state. This is the 3D structure of the wrong tautomeric state of the same trigonide metformin we know very well. Then what about this? This is 1 5-hydrogen shape. The energy required is only 2.7 kilocalories per mole. The 3-hydrogen shape requires 38 kilocalories. 38 kilocalories process never happens, and 2.7 kilo process always happens. So these are the details that he listed after another 3 months. After having done that, look at the surface. Look into the molecule, look at the surface also. You all do this all the time. When you look at your teacher, whether the teacher is willing to talk to you or not, you will come to know. If your teacher is having smiling face and is happy face, You would like to go to close to your teacher and you talk to him happily. But if you see the teacher is serious, if his project is not accepted, he is unhappy. On that moment, you don't want to go to him. Right? You want to wait for another 1 hour, then only you meet him. Exactly, every drug has got a surface property. Every drug has got a smiling face. Let us look at the smiling face of every drug. This is bicarbonate number 1. This is right tautomeric state. See this smiling face. The wrong tautomeric state. Look at the surface of the wrong tautomeric state. Wrong surface property. This is what always we have been writing for the 50 years. Look at the surface of the wrong tautomeric state. It is absolutely wrong surface. So we are not only thinking at the wrong structure, we are also considering the wrong surface of the molecule by ignoring the tautomerism. Now look at this molecule. This is a protonated bicarbonate. Protonated bicarbonate, metformin is always protonated. When protonated, the smiling face is not lost. This is deprotonated state. Again, smiling face is not lost. In all these cases, BG1, BGP1, BGA2, which are chemical processes, this is not facing, this is not changing its surface property, whereas wrong tautomeric state is a wrong surface property. So that means if and only if we can look at the surface, we come to know whether it is complementary to macromolecule or not. This is macromolecular surface, this is the molecular surface. This complementarity, you see, red versus blue, electron rich center versus electron deficient center. Complementarity becomes very clear. In the wrong tautomeric state, there is no complementarity. So small molecule, macromolecule interactions happen on the surface of it. So exactly we need to pay attention. Look at this molecule, which we all of us know. This is nothing but cycloguanyl, which comes out of the proguanyl. But if you go ahead and see, just don't do any research. Simply do zero research and rewrite the structure in the right tautomeric state. Look at the pharmacophoric features. These pharmacophoric features are more easily comparable than these two. Okay, so that is the reason why this paper was published in Journal of Medicinal Chemistry. You are all reading Journal of Medicinal Chemistry papers. Is there any paper you have read without doing organic synthesis published? Did you read any paper without doing any biological experiment? Day-2022 published. This was done only by doing pharmacophore mapping. No biology, no chemistry. Day-2022 paper. So that was the work of Mr. Dillon Patel. You know him?
Yes.
He was your.
I used to teach these papers to students also.
Okay. So Dillon was your senior?
Senior.
Your batchmate. Okay, Dillon Patel. So his classmate did this work. It's not, he was master student like you only. You can do, it is like if Dillon have done as master student, that means you have done as master student this kind of work and published in this kind of journals. Now, after another 4 years, Mr. Dillon suggested that metformin does not have covalent bond here, it has got a coordination bond. And that is a big story, one more big story. Based on that, we already published more than 30 research articles starting in 2009. Now this is about coordination chemistry between P and carbon, and this is about lone pairs. Looking into that, this is a P type of lone pair, this is a sigma type of lone pair, and that is chemistry. And then looking into look at the inversion barrier. Inversion barrier is about 9 kilocalories. Rotational barrier is about 5.4 kilocalories. That means after protonation, metformin starts jumping and dancing. It is like a person who got a job. A person gave an interview waiting for his results. 15 days, 3 weeks, result is not coming. He was dull, depressed. When the result will come, he got the result, he got selected. He got selected in Pfizer as R&D scientist. He starts jumping and dancing. Just like that, metformin, when it gets protonated, it starts jumping and dancing because its rotational barrier is only 5.4 kilocalories per mole. So metformin, actually forget it, metformin is never a drug. Metformin hydrochloride is a drug. There is a large difference between it. Now we look at molecular orbitals. This is cyclogonal. Its molecular orbitals are like this. This is a p type of molecular orbital. This is the sigma type of molecular orbital. All these details were published in 2011. Later on, look at this particular figure. It says that how progonal is getting converted into cyclogonal. This chemistry is different from the organometallic chemistry we do. In regular Suzuki coupling or Chan-Lamb coupling or Buchwald-Hartwig coupling, we believe that metal is causing the coupling. But here, cytochrome has got a metal iron center. Iron center is not causing the coupling in progonal to cyclogonal conversion. You all know about this reaction happening in 1942. It was identified. But who is doing, how is it happening? This was identified in 2014 only. By another scientist who belonged to Uttar Pradesh. He was citizen of Kanpur. He came to NIPER to do some research and he found that progonal to cyclogonal conversion does not happen with the help of the metal. Metal center is removing one H radical and removing another H radical and preparing a triazatriene. Triazatriene is unstable, that's why it is getting cyclized. That's the secret that we could identify and publish this. And then look at other molecules. There are many other molecules belonging to that class with coordination bond. Having done that, the question was, is there any other drug molecule which has got coordination bond between carbon and nitrogen? Yes, this is amantadine. You all know about it. H2 receptor blocker. Probably this is available in nearby chemist shop in maybe 1 tablet, 5 paise, something like that. Such cheap. This is antacid. This is another antacid, amantadine. All these, both these molecules have got carbon-nitrogen coordination bond. This is fungicidal activity. This compound is having fungicidal activity. This is having neuroprotective agent. All of them have got a carbon-nitrogen coordination bond. If that is true, can we design new molecules? Is it only concept which we should remember, reproduce in the exam or should we design new drugs? We have to do that. So that is what was done. And this is the pharmacophore. Using this pharmacophore, synthesis was done. This is called guanine thiourea, GATU. This is guanidine unit, thiourea unit. They collaborate with these two. Once this molecule is considered, this molecule carries that compound called nitrione character with carbon-nitrogen coordination bond. Having identified that, how to synthesize such molecules. This synthesis is not difficult. Medicinal chemistry people should not do difficult synthesis. We should do very, very low quality, cheap quality research, but cheap quality product. Product should be cheap and best so that entire public can receive it. We don't want to synthesize very costly products. We want to use very easy synthetic procedures so that we can produce cheap and affordable products so that what is the chemistry? Simple, treat with HBr, you get a bromo derivative, you condense with this, you get the product. Not a big chemistry here. This chemistry can be done in one day. You can synthesize 3-4 compounds in this route. Okay, that is what exactly was done. 3-4 different routes were identified. Doubly gonal thiourea derivatives like this or singly gonal thiourea. Many of these were synthesized. And then, having done that, further quantum chemistry was done. Further molecular orbitals were looked at. Coordination chemistry. And then further synthesis. Look at this kind of molecules called nitriones. These were synthesized in NIPER using this kind of multi-step, but each step is not so very difficult because medicinal chemistry should never do difficult chemistry. Should always do simple chemistry. Okay, you get the compound, you prove them using X-ray diffraction, and further do molecular modeling. Further, you do analysis, synthesize more compounds. Start with this thiol derivative, methylate, another methylate, then condense, dimerize, you get this compound. This is also another simple chemistry. Okay. Likewise, one can design and synthesize many compounds. This is what was done. This was done by computer-aided modeling experts synthesizing molecules. But all our organic chemistry people are very busy. So why don't we do synthesis? Modeling people can also do synthesis. That's what exactly we have done. And we did docking. Inside the docking, it became clear that these synthetic compounds can indeed show anti-malarial effect with reference to PFDHFR. Now I want to spend a little more time on this slide. What happens inside PFDHFR? It's a beautiful story. If you look at it in 3 dimensions, that story becomes clear. The cavity of the PFDHFR is negatively charged cavity. Positively charged terene derivative bonds get inside. On the other side, nearby side, there is NADPH binding site. NADPH is binding, these two binding sites are projecting towards each other. They face each other. What happens? The NADPH is willing to supply H minus to the other ligand. This small change is happening. H minus is going from NADPH to the ligand, inhibition happens. This is how inhibition is happening. So that is all. What is the story? So if our molecule, if my molecule, synthesized molecule is efficacious, that means the NADPH cannot submit to, if this process will be stopped. If this process is to be stopped, that's all. That means NADPH should not be utilized. If my molecule is applied and NADPH is not utilized, that means my molecule is satisfactory. If NADPH is utilized, even after applying my molecule is useless. This is a simple yes or no answer. So using that answer, we can do in vitro study. That is what was suggested to the in vitro people, and the same in vitro experiment was done. Before doing, molecular dynamics was also done. Molecular dynamics analysis was showing that, look at this. This is the experimental data. Experimental data was suggesting that human DHFR is not inhibited by this molecule, but PFDHFR is inhibited by this molecule. That is selective inhibition. But molecular dynamics is also suggesting the same. When using this molecule, if you block the human DHFR, the energy released is only 22 kilocalories, but PFDHFR, it gives 34 kilocalories energy. That means the binding affinity of the newly designed molecule is much larger in wild type PFDHFR, in quadruply mutant PFDHFR, doubly mutant PFDHFR, as well as another doubly mutant PFDHFR, but it is selective against human DHFR. That means it gives confidence that if I give a tablet to human will not be disturbed significantly. Malarial parasite DHFR will be affected. That means this is going to be a drug. Okay. So this is further molecular dynamics to show which amino acid is important. Here this cysteine, this aspartic acid, this cysteine, this arginine, they are important. Now you go to the in vitro laboratory. When you go to the in vitro laboratory, what is the experiment to be done? Just to see whether that hydride ion transfer is happening or not happening. You supply your molecule and check whether NADPH is utilized or not. At about 350 nanometers, you will get the signal. Fluorescence spectrometry, FRET experiment you can do. Whether at a particular frequency signal is appearing or not appearing. Yes or no. Again, on that basis, we can suggest that as a result, what happened? One About 36 compounds were synthesized. Out of that, 23 of them are showing in vitro activity. 36 versus 23. So ask any company, if 23 compounds are showing in vitro activity, when your synthesis is only 36, they will be very, very, very happy. Okay, that is not sufficient. Go to in vivo activity. Out of the 23 compounds, 6 compounds were showing anti-malarial effect. 23 compounds into 3 mouse into 3 concentrations. So about 9 mouse versus 23 compounds. You perform the in vivo study. After that, it became clear that 6 compounds were showing good activity out of the total 36, which became 23. Now it became 6. Then you observe the happy life of the mouse. Is it, we observe after giving the compound, we observe up to 4 days, D plus 7, that means after giving 7 days, you observe, notice that in the case of 6 compounds, the animals are happily surviving. Then you don't stop observation. You observe up to 14th day. Observe up to 60th day. It was noticed that out of the 6 compounds, 5 compounds were causing recurrence of the malaria. So that means out of the 36 compounds, only 1 compound is useful. That is good enough. 1 compound is useful in vitro, in vivo, in silico, in quantamo, in everything. It is that means 1 compound turned out to be curative from this exercise after doing computer-aided homework. Do thorough homework in the beginning, and all your energy in homework. Then only you go to our chemical lab. Then after that you go to in vitro lab as well as in vitro lab. You find that these are the compounds which are showing a sufficient activity in vitro, IGVP value of 400 nanomolar, and then this kind of work has helped us to identify, first of all, new chemical bonding was identified. That means contribution to the fundamental chemistry. Then new chemical class was identified and many compounds were synthesized. As I told that this is one of my previous almost 5 to 6 PhDs got graduated. 2 more PhDs are currently working on the same topic called nitrogen chemistry, which was identified on Mohali campus. Then you synthesize many compounds and then try them in vitro, in vivo, and then you get, if you keep doing, you will keep getting publications. That is side effect. Publication is side effect. Okay, with this, I would like to thank all of you. And I would like to first thank my teacher. He taught me how to do research. He is currently in Institute of Science. He taught me in America, currently in Amsterdam. And then he was the one who identified this coordination chemistry between carbon and nitrogen. No, carbon nitrogen was our identification. He did carbon chemistry and taught me a few things during my very early days. And Chakravarti, he is one of the leading medicinal chemists of, and these are all the funding agencies, and these are the agencies which recognize our work. Included IBM recognize our work, more embassy also recognize our work. So this is sufficiently satisfactory if one great pharmaceutical company, Eli Lilly type company, as well as IBM recognize your work. That is good enough. I hope so. That should not stop us from working. This is a student. I told his name a couple of times, and these are all the others who continued and they continued a lot. This is our NIPER. Some of you have come and he lived there for some time, 5-6 years. And this is a guest house where you will be accommodated if you visit, when you visit. This is our library building, this is our administrative building, and this is our my office. You are welcome. Yes. Questions?
Sir, I have one query. Particular molecule, like as you have told that this will affect the plasmodium, not a human being. Is it useful for after the people those who are suffering from malaria, or it may be before that it can be used as a, like that block that interactions with the human being and the parasite?
This is much before. Much before synthesizing the first compound.
Okay.
Before entering into the organic laboratory, we can tell in advance whether it will inhibit, selectively or not. Which particular amino acid is responsible for selectivity, we can tell in advance. That is the level where computer-aided and bioinformatics and all these pharmacoinformatics has grown today to that level. We can talk about metabolism, toxicity, drug delivery, pre-formulation, preparation, and finally the drug action. All of them can be studied in the computer laboratory first before entering into any experimental laboratory. That is the power of pharmacoinformatics today, and I'm sure it is going to grow, especially because of pandemic. Most of us cannot enter into labs. For last 2 years, there were difficulties. But computer modeling research can continue to be done. That was just our pandemic kind of helped in the growth of pharmacoinformatics. Then,
Yes, sir. I have one question. Because a lot of time students ask regarding the computer aided drug design. One is the success rate, although we know that this was the traditional. And another is how to find out whether this design molecule will be acting against our agent. That actually sometimes.
yes, that is a very interesting question. It is not easy to answer that question. At the same time, it's not a very difficult question also. The reason is like this. Any science is science as well as art. You know very well, Lucknow is having expertise in music. So many experts came here. Naushad Ali came from NIPER, from Lucknow. Okay, लखनऊ वाले का म्यूजिक के बारे में बोलने का कोई जरूरी नहीं. Okay, so you know what it is. So, but you think everybody can produce a beautiful music just by learning Saregama? Just because they learned the first 2 lessons, can they produce exactly like that? Computer-aided drug discovery, or regular drug discovery, or biology, or chemistry, or any science. Just because we know the rules and principles of chemistry, we cannot produce great results. The great results will come when we apply the science as an art. Okay? We have to be immersed with it. We have to be associated with the science that we are doing. We have to appreciate, we have to enjoy. And there is a chance that we can come out with useful results out of it. So if you treat CADD as a technology, never you will get a result. If anybody participates with it, know the inside out, what are the limitations of the technology? We should, 90% of computer technology is limitations. How many limitations are there? Every science has got so many limitations. Even the Darwin's atomic theory was proven to be wrong. Periodicity was proven to be wrong. So many principles are proven to be wrong after 50, 60 years. Newton's laws was proven to be wrong on electrons. Newton said that electron cannot move until somebody hit it. But inside that molecule, electron is always roaming around. It's never static. Okay. So Newton's laws also were proven to be wrong. Then what is we are? We are so small. Maybe it is. Sometimes when we participate, when we treat science as an art, results will come automatically. My opinion, all those people who are successful in science, they treated science as art.
Message on science lesson.
Message on science.
So,